talk is only the technical paper in this section, so I hope that my talk enjoy you more or less. This work focuses on the development of the algorithm that can be used to quantify the information of the RNA signal. Specifically, we can analyze this information on a glass slide. We still preserve the information of the tissue context, which is different from the RNA sequencing. And this work basically is the collaboration between the imaging and algorithms in Santa Clara, California, and also the tissue research and early development TRED team in Tucson, California, in Arizona. RNA biomarkers can be widely utilized in the cancer diagnostic community. As you can see here from the plot, that is shown that the only 2% of the protein expression can be used to target of the region of interest, while using the non-coded RNA can be used as a marker that can identify some interested of the target of the tumor information. And basically, the RNA biomarker can be used using the universal nucleic acid biomarker, as can present in the form of the punctate signal. And each punctate signal can reflect to a single molecule of the RNA. And the technology of RNA in situ hybridization, or EACH, can be used as a method in order to localize the RNA sequencing of interest by using the nucleotide probe over here. It can be used as to label the complementary of the RNA in order to localize the specific RNA sequences in a portion of the tissue. And currently, there is a challenge that is still limiting of the technology that can be used to quantify the information. For example, is the RNA signal can present in a very tiny spot or discrete spot over here, and also is formed as a larger region, as shown in this example of the B-cell lymphoma of the kappa lambda. And therefore, in this study, we proposed to develop the digital pathology solution that can be used to quantify the gene expression while maintain the tissue context to enable the single cell analysis. Specifically, we proposed the automated image analysis framework that can report the total number of the punctate isolated signals and also the estimation of the total number that present in the signal aggregate. For the method, we first prepare the tissue. The tissue has been fixed and also cut, and we use auto stainer to stain a tissue. And basically, we obtain 31 slides for use in development consisting of this tissue type. And the slide has been stained using the protocol of the simplex and also duplex of each using this probe of the study. And the slide has been captured using a mounted camera on the microscope, and also use the whole slide scanner as shown here at a sample example of the whole slide image. And here is the framework of our study. We have multiple steps consisting of the pre-processing step and the main processing step. For the pre-processing step, we use the color deconvolution. Basically, we decompose the RGB image into an individual biomarker image channel. We use a well-known method of the color deconvolution based on this study. And here is the example of the dual rich stain of the kappa lambda. We use silver to detect the kappa light chain and the signal formed in the black color. For lambda, we use SRB to detect and use the purple chromogen. And for the nucleus, we use hematoxylin as a counter stain to stain for the nucleus presents as blue color. And after unmixing, we use the image channel in the next step of the isolated spot detection. The goal of this step is pretty much we detected the individual spot present on the slide by excluding the signal aggregates. First, we have the input image. We do some pre-processing and mixing, and in this example, we have the black image channel. And then we extract the large aggregates of the signal from the tiny spot of signal. In this step, we use the method of the difference of Gaussian or DOG to extract the small detecting dot from the blob signal. After we detect the tiny spot, we then find the position by inputting the annotation of the XY coordinate or coordinates of the position at the center of the spot. As a result, we can report the number of the isolated spot in a portion of the tissue. And then we also verify the accuracy of our algorithm. We use the 31 FOV that obtained from the 31 slides, having an expert observer to verify by having a total number of counts report from the observer and the algorithm. It's shown very well correlated with the number of counts in each FOVs of the study. And then we use the information, the coordinate that obtained from the isolated spot detection step in order to derive the signal features. We position the coordinate on the portion of the slide, and then we crop the image to obtain the image patch. And in each image patch centered with the signal, basically we're going to use the Gaussian fitting function to derive the related parameter of the Gaussian. For example, we can derive the standard deviation and also the mean of the signal to represent the characteristic of the dot. An individual dot has been derived, the characteristic of the signal feature, and then we can plot the histogram of the dots that we are present on the portion of the image. The feature that we use intensity, blurriness, size, and also roundness. And the definition of each feature, basically we use the 98 percentile to represent the intensity. Blurriness, we use the standard deviation of the Gaussian fitting parameter, and the size we compute the fluid at half maximum can represent the estimation of the dot size. And roundness basically is show how the comparison with the parameter for the perfect Gaussian function parameters. We use the concordant correlation coefficient, or CCC, as a represent of this feature. The output from the previous step of the signal features can be used to estimate the total number in the signal aggregate region. This is the most difficult part because of the crumb signal, pretty much very hard to estimate how many dots in there. Basically, we use the principle of the linear relationship between the summation of the optical density within the signal aggregate divided by the summation of the optical density of the isolated spot representation that we derive from the histogram. We can approximate some spot representation into the formula. As a result, we can obtain the total number of the spot within the signal aggregates. And to simplify the problem, to have like more accurate and also better visualization and in validation, so pretty much we have brought the concept of subregions. Basically, we have the black image channel that can mask it out, the isolated spot in the previous step. And as a result, we have the residual image that contain only the aggregate signal. And then we use superpixel segmentation method in order to segment or group the pixel that have uniform intensity. As you can see here, is in each subregion, we obtain similar intensity of the pixels. So by computing the number of aggregate within a small subregions, we can use to estimate better in terms of the accuracy. And here is the results of the total number that report from each aggregate signal. And also, as a result, we can also report a total number of, which is a combination of the individual spots and also the signal aggregates on the whole slide. In conclusion, in this study, we have proposed the framework that can be used to quantify the RNA signal by maintaining the tissue context and within the single cell resolution. And this proposed method can be applied as a potential tool to utilize in the clinical application. Thank you very much for your attention. I'm happy to take any questions.
Okay, show of hands for any questions. Okay, maybe I could, oh, we got one.
So thank you for an interesting talk. What is the potential for multiplexing in this context? Is there a way to wash off the stain and restain with a different RNA probe?
Thank you for the question. So you're asking about the staining of the duplex, right?
Correct, yeah. So say that you want to monitor multiple RNA molecules from different genes, for instance. Is there a way to do that in a feasible way in the same section? Or is it single molecule?
In terms of algorithm, we separate the image channel. So after unmixing, so we have the image has two information of the dual rich. For example, in this case, the black and purple, we separate the channel, and then we can process each channel separately. But at the report, we can, show the total number of counts for both stains.